I'm Sandip. I'm a postdoctoral researcher at the University College London. Although the work that I'm presenting today is work that I did before this at the University Medical Center in Groningen in the Netherlands. Um, and the overall idea is whether we can understand um, depression using the complexity of movement patterns. And so let me start um, with what major depressive disorder is. It is a mood disorder, and it's characterized by persistent feelings of sadness, hopelessness, loss of interest, among other symptoms. And it's known to be a leading cause of disability worldwide. And the idea is that it can, it's in fact known to be one of the uh, top five causes of disability worldwide and affects about 264 million people. Now, there are many diagnostic criteria that you would look to see if a person is suffering from depression. And one of them is a reduction in physical activity. And this is what is important for us. And that is what we will be sort of focusing on. Now, we know that the pure levels of physical activity is related to depression. Depressed people have less activity and more disturbed circadian rhythms than non depressed people, right? And we know that one way to measure this, especially nowadays, is using accelerometer units in smartwatches, right? So, like if you have your Apple Watch or if you have your MI Watch or something, and it gives you these small units which will tell you how active you are through the day. And the good thing about this is that this is measured with very low patient burden. So the patient just has to wear a watch and doesn't really have to say, for instance, answer questions every day and things like that. Now, the question we are interested in is whether beyond mean activity levels, is it possible that more complex measures, right? And by complex, I mean in terms of complex systems, whether more complex measures are representative of the this complexity of movement, right? And that's the question that we will answer today. And to talk about complexity or to understand complexity a little better, what are we going to use is we're going to use a measurement called as the recurrence plot, right? Now, what's a recurrence plot? It really is just the patterns of recurrence, or when things sort of well, for lack of a better word, recur in a time series. That is, so let me try and explain this a little bit. So suppose you have a time series, like you see in the upper panel over here. Hmm. So in this time series, what you have is you have a so you have the amplitude on the y-axis and the time on the x-axis. And what we're interested in is we're really interested whether a particular point will recur in the neighborhood of another point. So if you take the first point, we draw a little blue neighborhood around it, and then we check whether in future, right, some other point falls into this neighborhood. And if it does, then we go down in the same one and we put a black spot on it. So in this particular case, for the first point, you see that the fourth point also recurs in the neighborhood. So you will put a black point in the matrix 2, 4 and in 4, 2. That's what you see below and you see that in the blue shade. And similarly, you would do this for every possible point in your time series. When you do this, you have what you call as a recurrence plot, which gives you an idea of how the complexity of this whole time series is. Okay, so we sort of split up our recurrence plot inspired complexity questions into two, right? Which is where the broad overall question is what difference in recurrent physical activity does depression produce? And we can split this question into two levels. You have a group level question and you have an individual level question, right? So the question in the group level is whether these patterns of recurrences that I see in the physical activity, do they differ between depressed and non-depressed individuals, right? And this is at a group level. So if I take an overall recurrence plot of many depressed individuals and I try and compare this with a group of individuals who do not suffer from depression, is it possible that the recurrence plots show different characteristics in the depressed group and the non-depressed group? That's the first question. Now, the second question is a follow-up to this, which is if there is a difference at the group level, does this occur prior to an episode of depression at the individual level? Right? Okay, so for the first question, which is on the recurrent physical activity at the group level, what we did was that we tracked the activity of 25 depressed and non-depressed individuals. We tracked them for about a month, and we found that there were no significant differences in the mean physical activity. Right. So this was in the move study, which is what we use. In this study, what we found was it's quite interesting that in this particular case, the most observed quantifier, which is the mean physical activity, which is whether the depressed group of people will move more than the will move less than the non-depressed group of people. And we found that in this study, at least, there were no significant differences, right? And then we actually used our recurrence-based complexity measures, and we just wanted to see whether the recurrence measures are telling us in a very complicated way what the mean can already tell us. But we found that there was really no correlation between recurrence-based quantifiers and mean physical activity. What this suggests is that what your complexity measures or your recurrence-based complexity measures is telling you is something very new from existing measures, right? Now let me come to the results of this first half of the study. So what did we do? We checked the recurrence plot of physical activity in depressed and non-depressed group, right? And what you can see is I've plotted on the left on figure A. I've shown the recurrence plot associated with a non-depressed individual, and on the right with a depressed individual. You can immediately see that the recurrence plot shows significant differences. Right? There seems to be more order in the non-depressed individual than in the depressed individual. In fact, this is true. When we try and quantify this, we find that there were significant differences in the average and the entropy of the diagonal line distribution. So these are two of the most commonly used measures, which is which is the average and the entropy of the diagonal line distribution. Let me try and show you the histograms associated with this. Um, so the average, so these are the histograms. So you can immediately see that the salmon or orange one is a depressed and the blue is the healthy or non-depressed, how you want to call it. So the average diagonal line distribution. So we try to interpret this a little bit, and what we found that, or what we interpret this as, is that the average diagonal line distribution is associated with the duration of recurrent physical activity. So, and the entropy is associated with the diversity of recurrent physical activity. Let me try and take a minute to explain that. Uh, what that means is that non-depressed individuals, or let's take the case of depressed individuals because maybe it's easier. So depressed individuals seem to show a lesser duration in recurrent physical activity. So you can take a recurrent physical activity, like say cycling to work every day, or like swimming, or taking a walk in the morning, right? So these are things that will repeat every day, right? And we found that these repetitive physical activities are less in depressed individuals than a non-depressed individual. And the diversity of these, which means that the types of things that you do, right? So depressed individuals seem to show a lesser diversity of these. So for instance, um, if you take a walk every day, and if you take a if you cycle to work every day, and maybe every week you go to the gym, right? Now we seem to find that uh, depressed individuals show a lesser number of these activities. Now, this, of course, one could argue that this is not particularly surprising, but what is quite interesting is that this does not seem to correlate with just the mean physical activity, which is what you're interested in looking at. You often just look at how the mean activity changes for an individual, right? And so that does not seem to show any difference. So essentially, if you just measure how much a person moves a day, that does not seem to be as effective in finding out if a person is depressed or not, as much as if you look at slightly more complex measures in the terms of recurrence plot. And that's a takeaway that I want you to take from this particular bit. If you're interested in more details, this bit has been published earlier this year and that's the reference below. Let me go to the next bit, which is the individual level question. Um, to answer the individual level question, the study that I will be using, or the study that I used, was a trans ID tapering project. In this particular study, what did we do? We were following individuals who were tapering medication and were at an increased risk of depressive episodes, right? So individuals who were formerly depressed for and were tapering their antidepressive medication, so they were reducing it over the course of about 4 months, 6 months. And what we did was, now it's well known in literature that when you taper your antidepressive medication, that is one of the most at risk periods, right, for depressive episodes. So we chose these individuals, and we requested them to wear a smartwatch. We also looked at a bunch of other things. We looked at the heart rate twice daily. We did something called as EMA about 5 times daily, and so on. And we did this for about 4 months. And uh, as far as the accelerometry or the physical activity data is concerned, there are many interesting questions you can ask. And someone else in the group is looking at the mean the mean physical activity and the circadian rhythm shifts. Right, all these questions are being studied separately, and hopefully in the next few months, the manuscript will be out. So if you're interested, you can take a look. Okay, so this was the overall project that we used, and now I'll try and explain the methodology. And the methodology is using something called as the sliding window. So we take a time series of 8 weeks of actigraphy prior to depressive transition, right? So I don't know whether I said this. So actigraphy is just another word uh, for, well, it's not another word, but in this context, it just means physical activity. So we took 8 weeks of physical activity data prior to a depressive transition. So when an individual experienced a depressive transition, we took 8 weeks of data prior to that. And then, if you remember what I was talking about earlier about the recurrence plot quantifiers, so we estimated the recurrence plot within a window of 2 weeks. So we have a time series, which is 8 weeks long. And we take the first 2 weeks, we calculate the recurrence plot. And we calculate the parameters of the recurrence plot, like the diversity and duration, the average and the entropy that we talked about earlier, and a bunch of other things, right? And then we slide the window forward by about Q points, all right? And we keep doing this. So we slide the window queue forward by, let's say, 10 points, and then you check in the next 2 week window what is the recurrence plot, and then you calculate the parameters of the recurrence plot, right? And you keep doing this. Now, what do you have at the end of it? You have a time series of parameters, right? Because you're sliding your window forward, and you examine the time trends for each recurrence plot quantifier, right? So, for instance, on the left, what you can see um, is someone who experienced a very significant trend over time, right? And on the right, what you see is someone whose trend wasn't significant because it seems to go up and down, up and down. So it's not particularly, it is not as significant as the trend on the left. And that's what we're sort of interested in. We're trying to see when people have these trends and whether these trends precede a transition towards depression. Um, okay, so actually, let me then go to the results and then I'll explain the results. So these are the sensitivity results. So when you see a blue box, okay, let me explain this graph a little bit. On the y-axis, you have the individual number. So these is so each number 0, 1, 2, 3, etc. refer to one out of 17 individuals who experience a transition. And on the x-axis, I have the different quantifiers I'm looking at: the determinism, the laminarity, determinism upon laminarity, the average, which is what I talked about earlier, the entropy, the average and entropy of another line distribution. And what the blue boxes mean was that a significant trend was found, right? And that's quite interesting because we found that we can see that there are most of the individuals, except about, I think, five of them, 16, 1, 2, 3, 4, 5, yeah, except five of the individuals, everybody really seemed to show some kind of trend in um, in all quantifiers. No, sorry, in at least one of the quantifiers. And many of them show these trends in multiple quantifiers. Right? So so the sensitivity yielded was quite high. Right? Just maybe one more small detail, which is that the color that's shown is the depth of the color. The deeper the blue is, it means the more significant the trend was. Okay, but that's not the only question. There's another question that we need to ask, and that is a question that needs to address the plot above on the plot on the right hand side, which means when an individual does not have a transition, right? Can you still see that there is some kind of changes found, right? And this all seems to happen. So the first graph, this one, the sensitivity, will give you the number of true positives, and this questions like this would be when you look at the false positives. I don't know whether, no, unfortunately, I forgot to put on the slide on how we define the false positives, but let me tell you, however. So what we did was that we took the time series of the physical activity of an individual, and then we picked a random point in between, right? And we sort of picked the random point such that it corresponded to the timing that a transition was experienced in another individual. Um, the transition was experienced in an individual in the transitioning group, right? So the group which experienced transition, they experience transition at various times. We pick out all those times and we create a set of those times. And we pick out a random element from that set, right? Which is just timing in which one individual experienced a transition. And then we plant a transition in individuals who did not experience a transition. Okay. And then now I do the same thing again. So now what would I expect? I expect that because there was no transition, there would be no rises prior to, or rises or falls, changes prior to these fake transitions. But this unfortunately was not the case. This is what my specificity of false positive results are. And you find that a large number of individuals show false positives in their show false positives in their recurrence quantifiers. And it is, of course, if you look over here, you can see that everybody except one, two, three, four, five, six, seven individuals did not show any changes as we thought it would be, but many other individuals did show transitions, and that is, well, slightly unfortunate, but that's the way of research, I guess. So okay, so then let me try and explain these results a little bit, and let me see, what are my slides? Yeah, okay, I have a few slides on this. Okay, so so what we found, the results are these, okay, which is that there were significant increases, there were significant decreases that were observed. And what the significant increase and decreases suggest is that the deviations from a rhythm may be more relevant than the absolute value. That means that um, was that okay? So deviations from a rhythm, in the sense that, suppose you have some kind of durational diversity of physical activity patterns, right? Now, if you deviate from this normal rhythm, that seems to be more of an issue than just individually trying to pick out, oh, increases were observed or decreases were observed. Now, he found that 70% of individuals showed warning signals, which means these significant trends, in at least one of the quantifiers prior to a transition. Right. Overall, we found that our sensitivity was 39%. In individual quantifiers, also, it wasn't particularly great. It moved between 35 to 47%. And the next bit, which is the part I was talking about, is that 53% of individuals showed warning signals when none existed. So there were no warnings. Suppose we expected that known warning signals would show up, but 53% of individuals actually showed warning signals, right? And the overall specificity was 71%, which is, it's not bad. And varied between individual quantifiers between 59%, so some were particularly bad, to 76%, some were pretty good. Um, Now, there's an important question that we need to ask over here, and that is the question of what means what you often refer to at risk, right? Which is that all the individuals that we looked at were tapering their medication, so all individuals were at risk. So it is possible that while many individuals were not did not reach the clinical threshold to have a depressive episode, many of them would have actually been on the pathway, for a lack of a better word, towards depression, right? Um, so it's possible that what you're seeing is that the false positives that we're talking about, well, they might be overestimated, right? So this graph may is not really all that bad in that sense, is what we are suggesting. In that many of these individuals who showed many of these warning signals may be on the path towards I don't know, could have been on the path to depression in between, and that is what is being picked up by these quantifiers. Um, yeah, so that's what we mentioned over here. Now let me end with the takeaways.